This is the brief on the acetate problem in E. coli fermentation. E. coli is the absolute workhorse of industrial biotechnology. We use these bacteria as microscopic factories, but when we scale up production to massive industrial tanks, they start churning out a toxic byproduct called acetate that basically ruins the whole batch. First, let's look at this scale-up sabotage. Small bench-scale batches work just fine, right? But moving to massive 500 to 20,000 liter production vessels causes acetate levels to absolutely skyrocket. To put that into perspective, this toxic buildup literally stops cell growth in its tracks and slashes protein expression by a whopping 50 to 80%. Second, you might be wondering, why does this even accumulate? Well, it comes down to a phenomenon called overflow metabolism. When the bacteria gorge on glucose faster than their systems can process it, or when oxygen levels drop too low, their metabolism panics. They shunt all that excess energy straight into making acetate. Plus, the sheer stress of producing foreign proteins just adds to this crazy metabolic traffic jam. Finally, let's detail the toxic toll and the massive payoff for fixing it. Acetate is a multifaceted poison. It acidifies the inside of the cell, depletes its energy, and causes proteins to misfold into useless clumps. But a game-changing 1988 Shimizu insight showed us why this matters. If we control the process and keep acetate near zero, we can literally double the bacterial growth and triple the active enzyme yield. Mastering large-scale E. coli biomanufacturing is really all about keeping a tight leash on glucose and oxygen, so overflow metabolism doesn't crash the party. Crack the acetate code, and you unlock the true powerhouse potential of industrial biotech.